Hey guys, how are you? I hope you had a good weekend. I did. It was my birthday. So, we started this next unit talking about cell theory. So, we did 1.1, the first part, which is really just talking about kind of the upper level ideas about and the history behind how we discovered cells and how technology played a big role in that, because without the microscope, we really wouldn't have gotten that far. And if you think about it, you know, it took like 200 years to come up with a cohesive theory about cells. And we're going to continue that line of thinking today when we're talking about more recent advances in understanding how cells compose life. So now we're moving beyond just the fact that all life is composed of cells and that they look like tiny little circle things, right? Now we're going to really be talking about how you go from single celled organisms to multicellular life. And that's capable, or we're capable of that because of cell differentiation. So, just as an introduction, individual cells are able to carry out those 7 functions of life that you need to know. You should remember what those 7 are. If you don't, go back in your notes and look. But they can also work together to form more complex entities. Remember, you're still doing the 1.1 understandings, applications, and skills. So, this is understanding number 4. Multicellular organisms have properties that emerge from the interaction of their cellular components. So, when you think about levels of organization within a multicellular being, right, we're talking about cells. Individual cells together make up tissues. Tissues together make up organs. Multiple organs together make up an organ system, and put multiple organ systems together to create an organism. So, there's a lot that has to change when you're talking about an entity that exists just as this all the way to here. And it's really the mitochondria and the chloroplast that we can thank for that, but we'll talk about that in topic 1.5. Just understand that the basic organization that you need to know: cells, tissues, organs, organ systems, organism. When cells fuse together and they organize, they develop what's called emergent properties, meaning these properties are not there when the cells function on an individual basis. It's only when they work together that these properties or these capabilities emerge. So these are functions and properties that individual cells can't do. So if you think about a neuron, right, a neuron is only capable of sending and receiving signals, but when you put them together, they form a network that's capable of producing consciousness and thoughts. And that's pretty astounding. Take one little dendrite on its own and it can't really do much. But when you have an organized system, you can produce something as complicated as consciousness. So in biology, scientists usually describe emergent properties by saying the whole is greater than the sum of its parts, right? So you have lots of different parts of cells, and we'll talk about that in topic 1.2. We'll talk about the ultrastructure of cells, so all the different organelles, just like we did freshman year. But the cell as a living entity on its own is greater than just those individual parts. Well, similarly, congregations of cells, right, and the different types of cells that compose life are greater together than individually. You don't get that emergent behavior until you have collections of cells that specialize to do different things, and that is differentiation. So, understanding number 5, specialized tissues can develop by cell differentiation in multicellular organisms. So, specialized tissues in an organism can develop by cell differentiation, which is the process of cells developing the ideal structures needed to carry out a particular function. Now, when we first started talking about cell theory during our last lecture, we talked about striated muscle fibers and how that muscle tissue kind of bucked the theory of traditional theory of cells, and that it had multiple nuclei per cell, which is unusual. So, that type of cell adapted to serve a particular function, right? It has specialized, it has differentiated. That's what cell differentiation is. You start with an individual cell, a stem cell, that can become any kind of cell, and it differentiates into a particular type of cell. That's differentiation. So, other examples, we have stem cells that become red blood cells, or that can become nerve cells or become muscle tissues. And these all have different structures and functions. So, these guys have they look like pillows and they have a little divot in the center to carry oxygen. These guys have multiple nuclei and they have particular proteins that help them stretch and contract. And these guys have a very unique structure. They have what's called the body and the arm. This is a dendrite or a neuron cell, right, that reaches out. And this particular form is adapted to transmitting electrical impulses. So, and all of these types of cells come from a stem cell, but the process by which they become these different things is differentiation. So there's 220 distinct types of cells that make you up, right? So 220 types of building blocks that make you. So we have bone tissue, which has its own particular structure and function. We have connective tissue, nerve cells, red blood cells, smooth. I'm not going to name all 220. That would be dumb. But you remember, life starts as the union between an egg cell and a sperm cell. So from these two individual cells that fuse together to become one singular cell arises 220 different types of cells that are required to build a human being. So understanding number 6, differentiation involves the expression of some genes and not others in a cell's genome. So cell differentiation occurs as a result of changes in gene expression. This involves some parts of the genome being used, but not others, right? This is important to recall, though, that all cells in the body have identical genes. So this singular cell contains all the instructions to become any type of cell. However, it's destined to become a specific type of cell using only parts of those instructions. So this same cell, once it divides, could become a neuron based on what genes are expressed, or it could become an epithelial or a skin cell based on what genes are expressed. So the destiny of a cell is related to the expression of specific genes. Now, we don't technically talk about genes until topic 3, but I want to take a minute and explain what gene expression is. So expression of a gene means that gene is flipped on and you're seeing the result of whatever that gene does. So if you have 2 sets of genes, one from each parent, right? And think back to freshman year and you talk about dominant genes and recessive genes, right? Well, dominant genes will hide recessive genes. That's because the dominant gene is being expressed. So gene expression is when you see the result of a gene. One gene is functioning and working properly, right? Now, just because you have a gene that's not being expressed doesn't mean there's anything wrong with it. It just means it's not turned on. So you've heard me refer to genes before as kind of like the instruction booklet with all the instructions to build you. But a better analogy is like a giant switchboard, and each gene is its own switch. So those switches can be on or off depending on what cells you're talking about. So the type of cell or the destiny of a cell is dependent on which of those switches are turned on in that cell and which of those switches are turned off in that cell. So once cells have differentiated, they can further fuse and mature to form specialized tissues. Here we have a couple of examples where cells specialize and fuse to create muscle fibers. We talked about that a little bit already. So this looks kind of complicated. Don't freak out. I'm not going to test you on this MRF stuff, but I want you to understand the process. So you have what's called a mesodermal progenitor cell. So all that means is meso means in the middle. Dermal is like a layer of tissue, like epidermal, mesodermal, endodermal. All vertebrates are composed of 3 levels of tissue from the inside out. Middle is endodermal. That forms the alimentary canal from your mouth to your butt, right? It's like a tube that goes through your entire body. Then there's the mesodermal, which is the middle, and that becomes your muscles in your skeleton, and then there's the epidural, which becomes your skin. So these are mesodermal cells, progenitor cells. Progenitor means like first or father, right? So this is the first set of cells, those mesodermal cells. They're going to differentiate into what are called myoblasts. And you can see these look a little bit more like muscle cells. You start to see those tapered ends, right? And then they fuse together to create one long fiber called a multinucleate myotube or myotube. And then you put a bunch of those together and you get muscle fibers. So this is a good example of differentiation and determination and maturation in cells. So you don't have a muscle cell that just like poop, there you go, another muscle cell. That's not how that works. So you have your progenitor stem cells, which go through determination, right? And they become myoblasts based on what genes are turned on. And then those myoblasts fuse to form a multinucleate myotube. And then those fuse together to create muscle fiber. And the differentiation is the last step. So from here to here, and is mostly differentiation. And then from here to here is maturation, right? We don't really distinguish too much between determination and differentiation at this point. These are all, if you want to know what these things are, these are external signals. So different hormones, different proteins, different external factors around that cell are what trigger those switches to turn on and off and tell cells what to become. So let's take some time and talk about stem cells. Understanding number 7. The capacity of stem cells to divide and differentiate along different pathways is necessary in embryonic development and also makes stem cells suitable for therapeutic uses. So stem cells are specialized cells that have the ability to divide and differentiate into different cell types. Zygotes, the union of an egg and sperm, are the most potent cell because, remember, they're the original cell that became you. So they have the most ability or the most potential, the most potent potentiality to become any kind of cell. And you can see these are all different kinds of cells. These are muscle cells. This is red blood cells. That's a neural cell or neuron. This is fat. I'm not sure what those are. Those could be, looks like ovary or eggs. This, I'm not too sure what that's all about. It looks like corn. So there's different levels of potency or different levels of ability to turn into different types of cells. And you do have to know those different levels. So the first level is totipotent. Totipotent cells can become any cell type. They are the most flexible, the most potent. They can become whatever. Then there's pluripotent. Plura meaning plural. They can become most cell types, but some things are off limits. And then there's multipotent, which can differentiate into a particular class of cell. So your original stem cell, your zygote, is totipotent, meaning it can become anything. Then there's pluripotent, which can still become many different types, but the selection is fewer. And then there's multipotent, meaning within a particular cell class, that cell can become anything in that class. Now you can kind of think of it as like going to school, right? So like when you're in kindergarten and you're in elementary school, your future could be anything. You could be anything you want when you grow up. By the time you graduate high school, you kind of have a good idea, like, are you going to be a humanities person? Are you going to be a science person? Are you going to be a business person? Are you going to be an arts person, right? So you're maybe you've ruled out like being an astronaut, but your options are still open, right? And then when you go into college, you specialize into like specifically, or trade school, specifically what it is that you are going to do. So you can think of it that way too. So lots of potential up here, a little more focused here, still some choices. Then you specialize and you become that very specific type of cell that has those very specific properties to do that very specific job. Because stem cells have that flexibility to become different types of cells, there's a lot of potential to use it for medical therapies. So you hear about miracle stem cell transplant for different things, people with spine injuries or brain damage or all sorts of things. And the truth of the matter is that there is a lot of potential there, but The process isn't as easy as we think it is, and there's still a lot that we're learning about how to make this work properly. So, there's lots of capacity. We have proof of certain things being healed or improved through stem cell treatment, right? We know in the brain. Now, remember, it depends on what kind of stem cell. You can't use like pluripotent cells for this. Depending on the level of specialization, you might have to use different levels of stem cells, some of which are harder to come about, and we'll talk about that in a minute. We know for sure that the use of stem cells for brain injuries and neural problems, spinal cord injuries, is a thing. We know that that helps. We know that bone marrow transplants are a thing. We use those quite a bit. So we'll take stem cells from an individual or from embryonic sources to rehabilitate bone marrow so it can start producing white blood cells again in somebody that's immunocompromised or has leukemia. We know Crohn's disease, some people have claimed that treatments of stem cells work. We know multiple site cancer recovery rates. So after you've been treated for cancer, use of stem cells to help recover the tissue sometimes helps. Things like baldness, you know, that's kind of a cosmetic thing. It doesn't really have a lot of research behind it right now. We're kind of focusing on the more important stuff. We know that deafness can be cured. Some particular kinds of deafness can be cured with stem cell therapies because you can reform those cochlear hairs. We know some forms of blindness can be treated with stem cells. We can also use gene therapy to treat some forms of blindness. Other things we're still working on, things like muscular dystrophy. There's lots of trials right now. Diabetes. This is type 1 diabetes, not type 2. Type 1 is when your own immune system attacks your pancreas and destroys the cells that produce insulin, meaning you no longer have the ability to produce insulin. So stem cells implanted into the pancreas so that the pancreas can regrow and then has the ability to start producing insulin again. You can therapeutically treat or cure diabetes. Type 2, that's different. That stem cells don't work for that. That's straight up, you got to change your eating habits. So you can see lots of different potential therapies. Some tried, some we're working on. So, application number 4, the ethics of the therapeutic use of stem cells from specially created embryos, from the umbilical cord of newborn baby, and from adults' own tissues. So there are different sources of stem cells. Not all stem cells are the same. We have different levels of potency, and they don't all come from the same place. So embryonic stem cells are made by fertilizing eggs with sperm and allowing the zygote to develop. Now, we're not talking about like developing into an infant. We're talking about developing into a cluster of cells. These cells have the most unlimited potential and can differentiate into any cell type. So that would be totipotent. Right? And then those cells can be cultured and turned into whatever cell is necessary. Now, let's talk about the ethics behind this, right? There are ethical considerations about how much this should be done on a couple of different levels. So, there is the belief that conception is the beginning of life, right? And that is a valid belief of some people. That people are entitled to their beliefs. So, the creation of an embryo for the specific purpose of harvesting totipotent cells has some ethical concerns there, because that cluster of cells does have the potential to become an individual. Now, there are incidences where extra embryos are created on a regular basis due to fertility treatments. So fertility treatments occur when people can't have children on their own. So what happens is the woman is given a bunch of different hormones that causes her to produce a whole bunch of eggs at one time. Those eggs are harvested, they're fertilized in a petri dish, and then they're implanted back into the uterus to implant and become a fetus and eventually be born. And in that case, you're not just fertilizing one or two embryos. You're fertilizing 25, 50, 100 embryos. And then you're taking just a very small percentage of those and implanting them back into the uterus, because the human body can't carry very well multiple fetuses, right? So humans, on average, they have babies one at a time. Sometimes you have twins. Occasionally you have triplets, but not very often. So most of the time when you see triplets, quadruplets, more than that, that's a result of fertility treatments, because doctors implant more than one embryo hoping that at least one will implant, right? So the result is you get lots of these extra fertilized embryos that are then frozen for later use, or they are disposed of because they're not necessary. That is another source of embryonic stem cells that can be used. So when you hear about bans on embryonic stem cell use, they're also talking about banning the use of those extra embryos that have been created through fertility treatments. So that's where the ethical impact or the ethical discussion comes into play, because on one hand, should you be creating embryos specifically for the purpose of culturing stem cells? There's a serious ethical argument against that. Now, I could argue it both ways if I was made to, but I could make an argument against that case. The second part of that discussion is, okay, well, we have all these extra embryos that are created through in vitro fertilization processes, and they're going to be disposed of anyways, right? So those could be used for medical treatment if current scientific and medical policy allows it. Now, in different countries, there are different rules. When I was in college, President Bush banned all embryonic stem cell research, and that was one direction I had been planning on going. But once he banned that kind of like prevented me from going down that path, because it was going to be really hard to live in the United States and follow a career in research that focused on embryonic stem cells, because you couldn't do it. I would have had to have moved to a different country in order to study it. So just be aware there are arguments on both sides, and you should be aware of what those arguments are and what the concerns are on both sides. So, the benefits: embryonic stem cells are the most potent. They have almost unlimited differentiation in terms of their potential to become different cell types. They also, we know from research, have less chance of developing mutations. So sometimes when you use stem cells for treatments, you have a increased chance of genetic mutations that occur in those cells. We're not exactly sure why, but we know that you have less of those problems when you use embryonic stem cells. So this is a fertilized egg that was fertilized during IVF. It becomes a embryo, a mass of cells, basically a culture blastocyst, and then you can use those for therapy. Downfalls of using embryonic stem cells: there's more risk of those cells becoming a tumor. Again, not exactly why that is. And also there's the ethical concern about the fact that you're destroying what could potentially become a life form if left to its own devices in the proper condition. Now remember, these groups of cells were fertilized outside of a human being and could not come to be a human being unless they were implanted in a willing human being. So it's not just like if you left it on the counter, it would grow into a baby, right? It would have to be implanted back into a person to be allowed to develop in order to become a human being. And that's an important distinction. So you may have heard of cord blood. So if you have younger siblings that were born, your parents may have opted, or even your parents may have opted to save the blood and bank it from your umbilical cord. And that's because that blood tends to have a very high concentration of stem cells, which makes sense because, That's a baby. The baby's a new individual. There are cells that are still differentiating, right? So there's lots of cells. Now these are pluripotent. So this is the bottom level. So, or no, multipotent is the bottom level. So this is the second level, right? So they can't become anything, but they still have lots of flexibility. Now, when you're born, that blood can be harvested and then it's sent to a storage facility where it's frozen, and then it can be used later to help you recover from things like cancer or treatments for different ailments. We thought about doing that when my daughter was born, but at the time, the storage was extremely expensive. So we decided not to. But if you can afford it, it's kind of a neat option. So the stem cells in your umbilical cord, they're multipotent. Actually, we just said that they're pluripotent, right? So they're pluripotent, but it's a blood cell, so it's a hemocytoblast. You can just call it a umbilical cord blood cell. That's fine with me. And they can become all sorts of different types of cells, right? So these guys over here, these all are immune system cells. So if you ever have a disease that wipes out your immune system, or you ever have treatment for a disease that wipes out your immune system, like you have chemotherapy or radiation therapy and it wipes out your immune system, you can use those umbilical blood cells to help reboot and rebuild your immune system. These guys are also immune system cells. And these guys, this is a blood cell, red blood cell, erythrocyte, same thing. Mast cell, yeah, these are mostly immune cells. So your umbilical cord blood cells are used mostly for recovering your immune system. That's a cord bank, so that's basically what it looks like. It just looks like a bag of blood, and they just freeze it. So, benefits and downfalls. So, benefits of umbilical cord blood cells, much easier to obtain and store, right? They come from the patient, so they're genetically compatible with that individual. So there's no like possibility of rejection because it's your literal own cells. I don't know why this is a benefit. Limited quantity in umbilical cord. That should not be a benefit. That should be a downfall. There's only a limited amount. Downfall, storage can be expensive, right? Limited capacity to differentiate, so we're no longer talking about totipotent cells, so your differentiation, the ability to differentiate is decreased, and there's a limited amount. Okay, adult stem cells. So you can harvest your own stem cells once you're grown up. The problem is they're a lot harder to harvest. They're a lot harder to obtain. You have a lot less of them, and they have like the least amount of growth potential compared to other types of stem cells. So you can process them. Your body always has a line of stem cells in it because remember, your body is constantly forming new cells, right? Like you know how like your taste changes every 7 years. Well, everything in your entire body changes over a couple of times at least while you're living. So when your cells change over like that, they're coming from stem cells. So you have stem cells throughout your entire life. It's not like they go away. You just have less of them as you get older, and they get harder to find and harder to harvest. You can get them from the brain, blood vessels, skeletal tissue, in the gut, skin. You can get it just about anywhere. But there's not a lot of them, and the process is not so nice. You have to isolate the cells from the patient. So if you're going to do skin stuff, you have to treat the cells with reprogramming factors. So you have to trick them into becoming, like working backwards and becoming stem cells. You have to wait a few weeks. And then you have to culture them into whatever you want them to be, right? So the process is, because there's more steps involved, it's a lot more complicated, it's a lot less successful, and it's a lot more expensive. So benefits, there's less chance of tumor development with these cells, though. Why? We're not so sure. They're fully compatible with the patient because they come from the patient that they're going to be used on. And the removal of those cells don't kill the adult, which is true. Downfalls, they're a lot more difficult to obtain, and there's less growth potential. So no longer are we talking about harvesting cells that can become anything. They're pretty limited as to what they can become. So we're going to move on to application number 3 that talks about a real treatment that we use stem cells to treat pretty consistently and pretty successfully. And that's the use of stem cells to treat Stargardt's disease. You should know Stargardt's disease, and you should, on your own, you have to know one other named condition. So you have to come up with one on your own. I'll tell you about this one, and then you have to go find another one. So Stargardt's disease is an inherited condition that results in the degradation of the macula in the retina. So this is in the eyeball. It affects juveniles, and it can result in blindness. You can see there's actually a hole that deteriorates macular atrophy. There's a hole that degenerates around the macula. You can see these flecks where it's starting to kind of thin out and degrade until eventually it gets so thin it tears and you see this big hole. So this is your eyeball from the side, right? So this would be the front of your eye. This is the back of your eye. This optic nerve goes to the brain, right? The macula is the thing in the back, and this is where light focuses, right? So your eyeball is a giant lens that focuses light into a very specific place. And if your eye is the wrong shape, that's called astigmatism, and the light kind of goes like that and it doesn't focus and your vision looks blurry, right? So if you're nearsighted or farsighted, I think it's if you're nearsighted, they cross before the macula. So it crosses here, so everything looks fuzzy. And if you're farsighted, it's the other way, I think. Anyway, this is the macula. That's the point where light waves focus in the back of your eye. And that's where the image, right? That's where the image is identified or recognized, and then that information is transmitted to the brain through the optic nerve. So if this thing goes bye-bye, you don't really see. The rest of this stuff doesn't matter. So, what does it say up there? Stargardt's macular dystrophy causes macular degeneration, and it begins to damage both eyes somewhere between the ages of 6 and 20, although impairment, like actual impairment seeing, might not be apparent until later in life, between ages 30 and 40. So in 2010, the US FDA approved stem cell research for the treatment of Stargardt's disease. Retinal epithelial cells. Okay, retinal means eye. Epithelial means skin or outer layer. So retinal epithelial cells are grown from embryonic stem cells and then implanted. So that means these were leftover IVF embryos that were then used to create medical therapies to treat a disease in a different individual. Initial trials indicated that the procedure was successful, so the next phase of tests have been initiated. Last I heard, this was actually being used. I think they may have changed the source of embryonic stem cells to something else. I'm not quite sure if they're using multipotent cells from individuals or not. But by 2020, 200 million people will have been affected by this particular genetic disease. And you can see here an example of what, so this is what you should see. And as degeneration starts, you see that. And then as that macular degeneration, the back of that spot in the back of your eye, you basically come up with a hole in your vision. Another example of a disease that can be treated using stem cells is leukemia. Leukemia is a form of cancer that stems from white blood cells inside the bone marrow. The cancerous cells invade the body quickly because white blood cells travel through the bloodstream, so they basically have a highway to anywhere they need to go. And leukemia is fatal if left untreated. I had a good friend that passed away from leukemia 2 years ago. We had a girl at Robinson that had one of our soccer players, and she had leukemia a couple of years ago. If you have childhood leukemia, chances of survival after treatment tend to be pretty high. The adult version of leukemia tends to be a lot more lethal. So if you get leukemia much later in life, it tends to be pretty devastating and usually pretty lethal within 2 to 5 years. Anyway, so in normal blood, you can see these are white blood cells here, right? In people with leukemia, remember, cancer is the multiplication of cells out of control. So you get lots and lots and lots of white blood cells that kind of take things over. And those cells invade pretty much everywhere. So symptoms include weight loss, fever, and frequent infections because your white blood cells, you might think like you would have a super immune system if you have leukemia because you've got white blood cells everywhere, but they're not doing their job. So in addition to multiplying out of control, they're like also not doing their job. So they're multiplying out of control and they're not doing their job. So you get sick frequently because you basically have a bunch of immune cells that aren't doing their job. Lots of different really nasty symptoms. So chemotherapy is used to kill cancerous cells in the bone marrow, but that also affects the body's ability to produce healthy white blood cells, meaning you wipe out your immune system. So before treatment, stem cells are removed. Healthy stem cells are removed from bone marrow and frozen, and then after treatment, you're given your own stem cells back to help recover and recuperate your immune system. All right, and that kind of just shows you the whole process, right? So they cut through your bone, they harvest stem cells, they treat them and culture them so they multiply a whole bunch, so you have a lot more. And then after treatment, they're transplanted back into the patient, and they go from that pluripotent cell into red blood cells or white blood cells or whatever kind of blood cells your body needs. Okay, so we're going to pause here. Make sure you upline your notes, and you should be able to answer these questions. Make sure you're going back and taking notes for all the objectives for the different understandings and applications that we did. Okay.